Well, it's my pleasure to introduce Jimmie Ye for our first seminar for this year. Jimmie is an associate professor in the Department of Medicine and also the Department of Epidemiology and Biostatistics at UC San Francisco. And I think Jimmie's initial background was in computer science from UCLA. And then after that, I think he did a postdoc at Broad Institute. And his lab's research seeks to dissect the interactions between gene and environment in determining human phenotypic variation. He has been at the forefront of the push to population-level single-cell studies, and his lab develops single-cell high-throughput profiling methods, and in particular, they are pioneers in developing technologies and methods for scaling up single-cell studies to large cohorts. Jimmie integrates methods from multiple fields, including statistics, computer science, and population and genetics and molecular biology to study the role of gene and environment in cell type specific phenotypes. I'm very excited to hear what he has to share with us. So without further ado, I will turn the mic over to Jimmie.
Great. Well, Nancy, thank you so much for the kind introduction and the invitation to speak. Everyone can hear me and see my slides? Okay, great. All right, so yeah, so I'm going to tell you a little bit about some of the work that's ongoing in my lab around how do we utilize multiplexed multimodal single-cell genomics to understand complex autoimmune diseases, and also to showcase two vignettes on what I think are some pretty exciting new technologies on the horizon that could potentially allow us to interrogate complex multicellular systems in different ways. All right, and so here are my disclosures. So I'd like to start with sort of like this slide that I show a lot of my first year graduate students on why they should care about sort of single cell sequencing, even if it's not something that they're pursuing directly in their graduate studies. This is adapted from Stetson et al. In Nature Protocols in 2018. And so it's already somewhat outdated. But basically what this figure shows is this dependence, exponential growth in terms of the number of cells sequenced on the y-axis and time on the x-axis. And so for those of you who are geneticists in the audience, this should remind you of sort of a similar kind of plot that people have made for sort of the number of DNA sequences in some reference database over time. And whenever you see exponential growth like this in data collection, it's almost always driven by technological advances. And so for us, the story really started roughly in 2010, where individual cells were picked and processed through bulk RNA sequencing methods, originally published by Barbara Wold's lab from Caltech. And when I was a postdoc with David Regev at the Broad, the field really took off with the advent of these microfluidic devices popularized by Fluidigm initially that allowed us to process libraries from tens to hundreds of cells simultaneously. But I think most of us are now using some sort of massively paralleled single-cell sequencing approach, either by encapsulating cells in very small microwells, in thousands of wells per experiment, or more likely encapsulating cells in a droplet-based microfluidic device. And of course, the most popular one is commercialized by a Bay Area company called 10x Genomics. And so, just in terms of scale, the current number of cells that we're sequencing per experiment is still roughly around the same as what was sort of advertised initially in 2017 by 10x Genomics, which is on the order of thousands to tens of thousands of cells per experiment. And so, the way that these assays all work and the way I like to think about them is that you essentially have some sort of physical compartment that has a barcode associated with it. So I'm just drawing this QR-like barcode outside of each physical compartment. You can think about these as droplets or wells. And then a cell will be associated with the droplet if it's encapsulated in that physical compartment. In the 10x-based system, because it's single-cell sequencing, the process of encapsulating cells in droplets is governed by a Poisson process. For most droplet-containing cells to contain only a single cell, you in fact would need to produce many, many droplets that don't contain cells at all. And we sort of noticed this when we were, just sort of reverse engineer the system in 2017, that this seems like a major waste of reagents, right? A priori, 10x wouldn't be able to know which of these droplets contain cells, but they have to provide enough reagents to process all the cells. And so really the name of the game is it's try to figure out ways to hack the 10x system so that we can put more cells into droplets. But in a standard run of the instrument, like I mentioned earlier, what's advertised is about 5,000 cells, 10,000 cells output. And what you get is a matrix where the number of rows is equal to the number of cells sequenced, so 5 to 10,000, and the number of features are the number of columns in that matrix. So if you're doing gene expression, this could be 30,000 dimensional data. I'm going to show a number of plots throughout the talk where we're going to take a 30,000 dimensional matrix and reduce the dimensionality for visualization purposes down to 2 dimensions. Each dot will represent a cell, and cells that are phenotypically related to each other are going to be closer to each other in an arbitrary 2-dimensional space. Okay, so many of us have sort of like used these droplet-based single-cell sequencing approaches to interrogate complex heterogeneous cellular systems. My lab primarily studies immune cells, and one example of a project is that we used standard 10x sequencing to profile tumor-infiltrating lymphocytes in bladder cancer. And the very first thing we did in bladder cancer is just looked at the frequency of CD4 positive helper T cells and CD8 positive killer T cells in bladder tumor. These are all patients who are being treated with checkpoint blockade, which are these transformative therapies that are trying to reinvigorate killer T cells to better neutralize tumor. And what we were surprised to find in bladder cancer using just standard flow cytometry is that the majority of the cells infiltrating the tumor are actually CD4 positive. And based on that result, we performed a single-cell sequencing experiment using 10x. This is in a low throughput way, 1 sample at a time, 5,000 cells each, to try to examine the heterogeneity of CD4 positive T cells in bladder cancer. And what we found were things that we expected to see. For example, there were multiple populations of regulatory T cells, here annotated by the expression of IL-2RA or CD25 and FOXP3, which is the canonical transcription factor for regulatory T cells. What we were surprised to find, though, were there multiple populations of what appeared to be cytotoxic CD4s. And so these are CD4 positive T cells that appear to express the same effector molecules that you usually detect in CD8 T cells, like granzyme K, granzyme B, and perforin. And there's some literature on cytotoxic CD4s, especially in long-term cell culture or in chronic viral infections. Some people have sort of like chalked them up as artifacts of culture. And so we were curious as to whether or not these bladder cancer infiltrating cytotoxic CD4s that we sequenced ex vivo actually could neutralize tumor functionally. And so what we did is that we isolated either CD4 positive T cells, all of them, or CD4 positive T cells, where we now sort out the regulatory T cells and all we get back are the effector T cells, and co-cultured it with autologous tumor at a ratio of 30:1. So if you just grow out the tumor on its own, then there's some sort of low levels of death rate. And if you co-culture tumor with all CD4 T cells, that rate is a little bit higher. But then if you only co-culture the tumor with effector CD4s, you can see that the CD4 T cells can actually neutralize tumor at a much faster rate. And this is actually similar to CD8 T cells. Of course, we know that CD4 T cells recognize MHC class II molecules on the surface of their target cells. And so we were wondering if these cytotoxic CD4s also are killing T cells through recognition of antigens presented by class II molecules. And so we co-cultured effector CD4s in the presence of either isotype control antibodies or antibodies against MHC class II molecules. And what you can see is that where there's no difference between culturing effector CD4s in tumors without antibodies and with isotype controls, when you culture cells with antibodies against MHC class II molecules, these CD4 effector T cells no longer neutralize the tumor, right? So we have this theory now that these cytotoxic CD4s, at least in the context of bladder cancer, are killing the tumors in an MHC class II dependent fashion. And so, this approach, like I said, has been used by many labs, not just ours, to understand complex cellular mixtures. But what if we wanted to study a more complex disease, or we wanted to understand more subtle shifts in composition or cell type specific expression between individuals? And so just to motivate the need for sort of population scale single cell sequencing, my lab studies a heterogeneous multi-systemic autoimmune disease called systemic lupus erythematosus that disproportionately affect biological females. And one could sort of like hypothesize that if you actually compared a two-dimensional reduction of the circulating immune cell states in lupus versus healthy controls, we really don't expect to see brand new cell types. It's probably a subtle shift of particular sets of cells that might be more activated or transcriptionally slightly different from a different cell type. And so that's quite different from sort of, when we think about other uses of single cell sequencing to detect rare cell types in a mixture. And the second application is that, imagine that we collected data on many, many individuals and we wanted to ask, what are the genetic determinants of gene expression and whether or not, the genetic determinants may be cell type specific, which we think is an important mechanism for mediating disease associations. Then these effects on genetic variants on gene expression are probably very small, and you would need very large cohorts to be able to have enough statistical power to identify these associations. So for both understanding changes between cases and controls for a complex disease and for mapping genetic effects on gene expression, we think there's an opportunity for large-scale population-level single-cell sequencing. The question is, how do you do such a study? And so the way we've been sort of thinking about how to do this is we didn't, at the time, there was not a new piece of technology. We just designed a new way of essentially hacking the standard 10x genomics approach to increase the cell throughput and reduce the batching effect so that we can generate really great population scale single cell sequencing data. The approach is called MUC-seq. And the way it works is actually really simple. Intuitively, we hypothesize that there's enough genetic variation captured in the transcriptome of a single cell that if we were given some reference genotype, that we could actually correctly assign each cell back to the donor of origin. And so, it's a little bit scary sort of if you're a conspiracist, because if we do actually have large scale genotypes for everybody, it's true that there's enough genetic information contained in a single cell that we can uniquely call out that individual. And that's because you only need about 50 markers to uniquely identify somebody. And there's more than 50 markers in the transcriptome of a single cell. And so that basically motivated a simple experimental approach for increasing sample throughput, which is you just mix cells from individuals that are genetically different, and then you do single cell sequencing. And then we have a method that will, given a genotype or a set of genotypes, assign each cell correctly to the donor origin. But there's a second feature through genetic multiplexing where we could use the idea to increase the cellular throughput into these droplet-based single cell sequencing approaches. Remember, I mentioned earlier, you have a lot of droplets that are empty, but if we increase the throughput by stuffing more cells into the instrument, we will get more droplets containing cells, which will contain single cells, but we will also get droplets that contain multiple cells. And the insight we had is that if you do a genetic multiplexing experiment, the vast majority of doublets or droplets contain two cells will contain two cells from two different individuals. In fact, the more people you multiplex, the more likely that you'll get these doublets from two different individuals. So our algorithm can also identify cases where there's genetic information coming from two different cells from two different people. And then we can throw that data away post-processing. So we still get bona fide single cell sequencing data and remove some of these confounders. But of course, we can't do this forever because these cells are still sequenced and that data is not useful. And this is going to come into play later on when I describe a new approach where we try to resolve all of the information in these multiply encapsulated droplets. But the version of MUC-seq that we described in Nature Biotech in 2018 still increases the throughput of single cell sequencing by about 5-fold. So routinely in my lab, we sequence 25,000 cells per run of the 10x instrument rather than 5,000. Importantly here, sample information is automatically encoded. We showed in our paper, it's highly accurate and simple to do. We originally demonstrated the approach for single cell RNA sequencing. You can also apply it to single cell ATAC-seq or single cell multimodal sequencing. And statistically, it's important that these approaches can actually minimize batch effects so that we get much better data analysis downstream. Just a few quick words about multimodality. So in 2017, there's a method that came out of New York Genome Center where we can take an antibody and conjugate a DNA molecule to an antibody. Compared to other ways of doing surface protein profiling like FACS or CyTOF, conjugating DNA molecules to antibodies gives us an opportunity for much higher plex profiling. So instead of looking at only 4 to 16 markers like FACS or 50 markers using CyTOF, CySeq is upwards of 300 markers today that you could purchase through a commercially available vendor. And so you could potentially do multiplex single-cell RNA sequencing plus surface protein profiling through CySeq and have all of that across multiple individuals. So you can get multimodal information per individual. So I'm going to skip sort of like how that works in cartoon form, and I'll just mention that you could also do other types of multimodal profiling. For example, using the current 10x Multiome kit, which measures chromatin accessibility in RNA. Here, it turns out that you can actually do a much better job of demultiplexing and identifying doublets when you have chromatin in RNA, because there's additional genetic information encoded into regions of open chromatin from the Multiome assay. And so here, this figure basically shows a latent clustering of PBMCs from analyzing jointly the ATAC-seq data and RNA sequencing data. And then the middle panel was showing the different individuals, so individuals colored by their genotype estimated by ATAC-seq, and then individuals colored by their genotype estimated by RNA sequencing. And you can see that you're basically capturing the same individuals. So that's MuckSeq. Now, we built it, like I mentioned earlier in the motivation slide, to try to use it to study large population cohorts. And so we started doing that as soon as the method started to work. And our first data set that really demonstrates the feasibility of multiplex single-cell sequencing on large population cohorts is this data set that we collected in lupus patients. So here there's a total of 165 patients and 99 healthy controls that are age and gender matched. We sequenced 5,000 cells per donor, generating a total of 1.2 million cells. The cohort is about 2/3 European and a third Asian. And because lupus predominantly affects females, our cohort is mostly female. In total, we sequenced 23 pools, and each pool is sequenced on 4 channels of 10x. And for some of these pools, in addition to getting single-cell RNA sequencing data, these are for at least 2 of the processing batches, we piloted an experiment where we're getting both RNA and surface protein expression in a multiplex fashion. And so here's a projection of 1.2 million cells. Each dot again represents a cell. And since these are PBMCs, we know a lot about the expected populations we would see in circulation, and we certainly recover them, including multiple populations of myeloid cells, or non-classical monocytes, classical monocytes, 2 populations of conventional dendritic cells, plasmacytoid dendritic cells, and then this population that sort of transitions from CDCs to PDCs are the AXL-positive dendritic cells described by Villani et al. In Science in 2014. We also detect a small population of CD34 hematopoietic stem cells, as well as multiple populations of lymphocytes, including natural killer cells, 2 populations of CD8 T cells, they're naive and effector memory, as well as CD4 T cells that are mostly central memory, proliferating CD4 CD8s, proliferating natural killer cells, small population of plasmablasts, and of course, CD19 positive B cells. And if we now take all this data and color cells based on their case control status and genetic ancestry, what I hope you can appreciate is sort of this point I made earlier, is that there is this shuttle shift when you compare cells from lupus patients versus healthy controls. There's not a brand new cell type. It's just that in patients, for example, we're much more enriched for effector memory CD8 T cells versus naive. And it looks like some of the myeloid cells might be shifting in their distribution as well. And this is conserved for individuals of European and Asian ancestry. So that's qualitative. Now, before we do any further analysis, we wanted to evaluate how quantitative single-cell, multiplex single-cell RNA sequencing actually is. And I just want to quickly remind you of what we did here. We took frozen blood, 16, 20 samples at a time, thaw them, multiplex, sequence, get the genotypes, demultiplex, call cell types, aggregate. And now we want to get an aggregated sort of molecular or immunological phenotype for each sample. And so the first thing we can do is just group cells at a much higher level. Let's call all the 4 populations of dendritic cells and monocytes as myeloid, and then the rest of the adaptive immune cells as lymphoid. And so we can quickly quantify the frequency of lymphocytes versus monocytes, or lymphoid cells versus myeloid cells. And what we observed is a known feature of lupus, which is lymphopenia. So lupus patients have a lower proportion of lymphocytes and a higher proportion of monocytes. And here in the controls, you can see that there are 2 biological replicates for the controls are actually quite reproducible, which is indicated by a line across them. And so now we have our very first quantitative metric that we derive from this highly complex single cell sequencing data. And so we wanted to evaluate in an unbiased way how quantitative is this data. And that's actually what motivated this broad classification of myeloid cells versus lymphoid cells, because we can convert these percentages into absolute abundance, since we also measure the number of PBMCs per volume of blood, and then correlate absolute myeloid cell abundance and lymphoid cell abundance with what's reported in electronic health records of the same individuals 6 to 12 months ago using a completely different assay and completely different blood draw. And so when we do that, we were really surprised and pleasantly surprised by how high the correlations were between estimates from single cell data on the x-axis and estimates reported in the AHR on the y-axis. So for myeloid cells, if you remove these 2 outliers, that correlation is remarkably good. Even if you included the outliers, that correlation is R squared of 0.9. For lymphocytes, that correlation is even better. It's about 0.95 R squared. So this data is important for us to now be convinced that single cell sequencing in a multiplex way is actually really, really quantitative. Of course, what the real power of single cell unbiased profiling of circulating immune cells is that we can go beyond just looking at myeloid versus lymphoid percentages, but ask what's changing in the frequency of various different subsets of immune cells. And what we observed is this decrease in the frequency of CD4 positive helper T cells in cases versus controls, and then a concomitant increase in the classical monocytes. And I'll just note that there's actually no difference in the abundances of monocytes between cases and controls. It's just really a decrease in the abundance of CD4s that also, because when you're doing a proportional analysis, it's relative to the total number of PBMC sequenced. This increasing the frequency of classical monocytes is all due to a decrease in the abundance of CD4s. We also saw some increase in the frequency of proliferating lymphocytes in lupus patients. We also compared these changes in broad composition between individuals with different genetic ancestries. And for most of the cell types, it's pretty consistent between East Asians and Europeans. But for CD4 T cells, what we observed is that there's an even larger decrease in the frequency of CD4s in patients of Asian ancestry versus patients of European ancestry. And we do wonder whether or not this is actually a feature that contributes to potentially developing more severe symptoms or lupus symptoms in these patients. When we first saw these results, we also were concerned that since we're a tertiary provider at UCSF, many of our patients are on a number of different therapies. And so we wanted to check whether or not this decrease in CD4 T cell frequency is due to treatment. And encouragingly, it doesn't look like it. There's subsets of individuals of both East Asian and European ancestry who haven't been treated by anything, including steroids for the past 6 months, and they still had a decrease in CD4 percentage. And if we also separated out which treatment each of these individuals got, whether or not it's Cellcept or Plaquenil or B-cell inhibitors, there's almost no difference in terms of treatment on CD4 percentage. So we think this depletion of CD4s is a real effect of the disease. And if we subtype the lymphocytes even more, we can further identify that it's really the naive CD4 positive T cells that are reduced in abundance in lupus patients. And then we also observed an increase in some frequencies of effector memory CD8 T cells, in particular these granzyme H CD8 T cells that in some individuals are 50% of all lymphocytes. And so we wanted to examine those CD8 T cells a little bit more. The first thing we did is we really wanted to explore the sort of the high dimensional features of our single cell sequencing data to ask this question about shared states in CD8 positive T cells. In lupus patients, there's this known observation that many of the immune genes, which I'll return to in a few slides, have an elevated expression of type 1 interferon stimulated genes. And many have hypothesized that in lymphocytes, the high expression of ISGs are actually correlated with some of the cells adopting a more effector state. And so we wanted to ask within our CD8 positive T cells, if we score the cells using a cytotoxic feature gene set or exhaustion gene set or type 1 interferon stimulated gene set, are they tracking the same cells? And it turns out that they're largely not. So cells that are cytotoxic are not expressing type 1 ISGs, and cells that are exhausted are not expressing type 1 ISGs or expressing cytolytic markers, which one may expect. And so this type of data would be completely obscured if you did bulk RNA sequencing, because all 3 gene sets would appear to be higher in lupus patients. We wanted to get some additional data to maybe help us understand whether or not these CD8 granzyme H positive T cells could potentially be causal for disease. And the way we went about this is did TCR receptor sequencing. At the time, we only got 3' sequencing data, so we went in and amplified for the TCRs ourselves. Our recovery wasn't very good, but we had enough cells still for us to get an estimate of the repertoire in cases versus controls. And we measured the diversity of repertoire using a normalized Shannon entropy. So the lower the normalized Shannon entropy, the less diverse the repertoire is. And so in lupus patients, the CD8 positive T cells are more restricted, so they're less diverse than in healthy controls. And we think that this could be because some clones are just being expanded. And to actually measure this, if we look at the proportion of clones that are expanded CD8 positive clones, most of them are in fact coming from the cytotoxic cluster of CD8 granzyme H positive T cells. So far, I've talked about sort of broad compositional changes. We saw depletion of naive CD4s and increasing the frequency of naive effector memory CD8s. I mentioned earlier that there's this other feature about lupus that's been known for about at least 2 decades, where circulating immune cells from lupus patients appear to be expressing genes that respond to type 1 interferon stimulation, which are normally seen in acute viral infections. And so we wanted to assess whether or not we see this as well. And the first thing we did is we treated our single cell sequencing data as though we had done a bulk RNA sequencing experiment. We just aggregated all of the counts, normalized them properly, and plotted them based on case control status. So here, each column is an individual. The red are the cases, and the green are the controls. And we identify a number of genes that are differentially expressed between cases and controls. And in particular, I want to call your attention to this black cluster of genes and this blue cluster of genes that appear to have pretty much the same pattern between cases and controls, where about 50% of lupus patients have high expression of ISGs and the other 50% don't. And so this is consistent with what others have reported, that this particular feature is not observed in all lupus patients, but certainly is very specific to lupus patients and not observed in healthy controls at all. And so that recapitulates what's known, but what's great about having single cell sequencing data is now we can ask, what exactly are the cell types that's producing these signatures? And when we did that, we observed that there's actually a difference between these black genes and these blue genes. The black genes are what we're calling a Penn up cluster. So these are genes that are upregulated in all major cell types that we profiled. And then the blue genes appear to be very, very myeloid cell specific. And they're enriched for the complements, for example, C1QB, as well as RNA editing enzymes like ABLBAC3A. And so this type of information, again, can only be recapitulated from single cell RNA sequencing. But something else we could do is now we have changes in cell states in specific cell types, like, for example, here, an elevated expression of type 1 interferon stimulated genes in myeloid cells. And then we also observed earlier some changes in composition, for example, a reduction in the frequency of naive CD4s. When we have data from this compositional data and changes in cell state from the same individuals, we can start correlating these immunological compositional features versus molecular features on gene expression. And when we did that, we were pretty surprised to actually initially find this inverse correlation between the elevated expression of type 1 interferon stimulated genes in myeloid cells on the x-axis versus the frequency of naive CD4 T cells on the y-axis. So the more activated your myeloid cells are, the fewer naive CD4s you have. And I want to say that these were all carefully normalized because, of course, I mentioned earlier that the frequency of myeloid cells is actually higher in cases as well. So this is independent of frequency of myeloid cells. But we think that this feature, and one more point here is that this is observed in both patients of European and East Asian ancestry. And we think that this feature is actually consistent with the pleiotropic effects of type 1 interferons. Maybe it's not surprising that type 1 interferons actually activate ISGs in myeloid cells, maybe preferentially than other cell types. But perhaps slightly less well appreciated is this effect of type 1 interferons originally described in mice by Jason Sister and others, where elevated type 1 interferon levels can actually lead to sequestration of CD4 T cells in secondary lymphoid tissue through the upregulation of CD69 and S1PR1. And so we think what we're seeing in circulation, at least in humans, is that it could be a proxy of CD4 T cells being stuck in tissue, and thus there's a reduction in naive CD4s in circulation. So a lot more work needs to be done to really prove this, including sequencing the actual tissue, but we're pretty excited about, I think this is sort of like the first time anybody has observed this effect that was originally described in mouse in humans. This is a fairly large cohort that we sequenced. In total, it was about 250 individuals. It is a case control cohort, but we also wondered if there's enough information here for us to start to do a simple genetic analysis where we can map genetic control of gene expression across all of the major cell types. And we started to think about this problem, and we did a vanilla analysis. What we noticed is that most immune cell types shared a lot of the same genetic controls. And that's not so surprising if you sort of think about the results from the GTEC consortium across much more diverse cell types. And so we wondered if there's a way, given the single cell nature of our data set, that we could, in fact, decompose a gene expression into a pleiotropic shared component across all cell types, and then a residual cell type specific component, and then use the shared component and the cell type specific component as individual molecular phenotypes for genetic mapping. Okay, so this is a method called FastG/C that we developed with Brunilda Balil and Noah Zeitlin at UCLA. And the goal then is to identify truly cell type specific genetic control, as well as genetic control that might be shared across cell types. And so when we use these two different sets of traits to map for EQTLs, we identified, not surprisingly, almost for every single gene that we had robust counts at the gene expression level, there appears to be a genetic variant that's associated with its expression in cis. And so here cis is defined as plus or minus 100 kb from the transcription start site. But when we also look at the cell type specific residual component, we observe some additional associations, particularly in the classical and non-classical monocytes, and they appear to actually have a very similar profile across the genome. And then also CD4 positive and CD8 positive T cells, of course, these are related lymphocyte populations, then they also have a very similar sort of profile across the genome. So how do we know that these cell type specific cis-EQTLs are real? What we did is compare the location of where the cell type specific cis-EQTLs are with regions of open chromatin that are cell type specific. And that data we obtained through a paper that was published by Calderon et al. In Nature Genetics, where they sorted for B cells, T cells, natural killer cells, and myeloid cells and perform ATAC-seq in bulk. And so cis-EQTLs that are B cell specific in our data set are enriched for B cell specific attack peaks. And this is also true for the T cells, the natural killer cells, and myeloid cells. So this result gave us confidence that what we're detecting as cell type specific are actually enriched for cell type specific cis regulatory elements. And is this exercise at all useful for interpreting disease associations, right? I think one of the conundrums in human genetics is that we know that there's cis genetic control of gene expression, but if you look at the overlap of GWAS loci with EQTLs, usually that overlap is statistically significant, but pretty underwhelming. And so one hypothesis is that many genetic variants associated with disease, in fact, have context-specific effects, maybe only active in specific cell types or in response to particular perturbations. And so using our data set, we assessed whether or not cell type specific cis-UQTLs could be more enriched for diseases. We analyzed 7 different non-immune related diseases and then 9 immune related diseases. So I just want to call your attention to a few of these. So Alzheimer's disease, which is actually a non-immune disease, is of course known to have these genetic associations that appear to be in microglia, which look very similar to innate immune cells in circulation. And so it's maybe not surprising that we see this enrichment of cis-UQTLs detected in classical monocytes, non-classical monocytes, and PDCs amongst Alzheimer's disease GWAS associations. If you look at lupus, again, the enrichment appears to be strongest for classical monocyte cis-UQTLs and cis-UQTLs detected in B cells. Again, these are, I think, results are consistent with what we know as the pathological cell types in lupus. And then one last result is if you look at the shared component, so these are genetic associations detected in all cell types, they are overwhelmingly not enriched for disease associations. And so I think this lends evidence that many disease associations are in fact cell type specific associations and are not the genetic variants that are detected across all cell types. I just want to show you one example of a locus that I think is of particular interest. It's around this gene called ORMDL3, which is actually very highly associated in asthma. But in the latest lupus GWAS, there's also a hint of association in the locus. Now, it's a difficult locus to study because there's 3 genes, IKZF3, gastrodermin B, and ORMDL3, that are all implicated to have some sort of immune function. And so it's been hard to identify what are the causal genes mediating this association. If you look at our cis-UQTL data, if you look at the shared component, there's genetic association to ORMDL3, which is highlighted in red, and then also to gastrodermin D, but there's no association to IKZF3 or gastrodermin A, which is another gene a little bit downstream. But the signal is strongest to ORMDL3 expression and not gastrodermin B expression. But furthermore, in specific lymphocyte subpopulations, in particular B cells and CD8 T cells, but surprisingly not CD4 T cells, there's an additional association to ORMDL3 expression that's detected in B cells and CD8s, and it's not observed for gastrodermin B. And so here on the right is just the raw data that we use to estimate this effect. All right, so that's the lupus study, and I just want to sort of give a shout out to the data. We actually have deposited this data in the Chang-Zuckerberg Initiative Cell by Gene Matrix months before the paper was accepted in press and now in Science. And so if you want to play with this, feel free to hit this link and look up your favorite gene. And there's also extensive annotations on metadata for the cohort that we profiled. In the interest of time, I'm going to skip the COVID stuff and just briefly touch on some of the other activities related to the application of MUC-seq to large population cohorts. So I described the lupus work. We also did use our multiplex single cell sequencing approach to look at type 1 interferon deficiency in critical COVID patients, generating 600,000 cells where you also have multiplex site-seq data. This is also available on the CZI website, so you can play with that data set. We're sequencing a number of cohorts in cancer immunotherapy and also in autoimmunity, in particular for Sjogren's disease, which we're very interested in because of the shared genetics between Sjogren's and lupus. But what about healthy variation, right? Some of the effects that we're seeing could just be due to variability across individuals, may have nothing to do with disease. And so to better understand what natural variation look like in healthy controls, we're actually performing a study where we're sampling 400 individuals across 4 ethnicities: East Asians, Europeans, African Americans, and individuals of Latinx ancestry. And we have now finished generating that data where we have multiplexed RNA sequencing as well as ATAC-seq data. And we're hoping to sort of analyze that data and put the paper out on Bar Archive, hopefully sometime this year. So look out for that. All right, so for the rest of the time, I just want to do sort of 2 quick vignettes on technologies that we've developed in the last year or so that we think creates brand new opportunities for functional genomics and proteomics. And both approaches are basically based on this idea called combinatorial indexing, originally popularized by Jason Dury and Claude Trapnell. But the goal of what we're trying to do is basically scale single cell sequencing by another order of magnitude as we go into sort of like 5 years since the original 10x platform came out. So why would we want to do this? I think for population scale single cell sequencing, throughput is important, but also for massively parallel screens where we want to do either drug screens or genetic perturbation screens, increased throughput is going to be useful as well. And so here's sort of like the intuition for what I'm going to describe. The first approach is called CytoSeq. Basically, what we do is 2 rounds of combinatorial indexing so that the end goal is to make a single run of a 10x instrument productive in a sense that every single droplet will contain at least 1 cell in it, which means that sometimes you'll get 2, 3, 4, even 5 cells. Remember what I said earlier is that if you do genetic multiplexing, when you have multiple cells, you might be able to detect them, but you have to throw that data away. With combinatorial indexing, we can resolve all of the data. And so that 4 or 5 cells in a single droplet can be resolved and you can get molecular profiles for each cell. So how does that work? So we built this particular version where we're only focused on doing surface protein profiling. So similar to sort of CytoSeq. And the way it works is that we're going to make antibody panels where each antibody will be conjugated by a DNA molecule that's specific to the antibody, and which is very similar to CytoSeq. But then what we do specific to CytoSeq is we're going to use another DNA molecule that will hybridize to the antibody specific molecules and then add a barcode that creates sets of these antibodies, right? So that first set will contain a purple barcode, the second set will contain orange barcode, and then the third set will have a brown barcode. And then these sets of antibodies are used to stain cells in separate pools. So here we've generated 3 different pools of cells that are stained. And then the stained cells are loaded into a droplet-based sequencer like the 10x instrument, and now much higher loading concentrations where we could easily have multiple cells in a droplet. And the idea is that cells that are in the same droplet will have the same droplet barcode, but they will very likely have different initial rounds of pool barcodes. And so that the combination of the pool barcode and the droplet barcode will allow us to resolve all of the data. So the analogy I like to use is the birthday analogy. So you can imagine that first barcode is the month of the year that a cell is born in, and then the second barcode is the day of the month that the cell is born on, and then the combination will give you a unique birthday for each cell. So how does this work, or does it work? Here's a pilot experiment we did where we took a 28-plex antibody panel where we conjugated ourselves, and never thought that my lab would be doing any protein chemistry, but we did our own conjugations, attaching DNA molecules onto antibodies, and sequenced in one 10x experiment, we pooled 200,000 cells into it. And we got back 50,000 cell-containing droplets. And we think this is not the limit of what an instrument can do, is that you'll have 50,000 productive droplets. And so if you look at all these 50,000 droplet barcodes and try to analyze the data, there's no information. It's just a blob. You can't tell CD4 T cells from CD8 T cells or myeloid cells or B cells. But if you now use the pool barcode, and there's 10 pool barcodes, in addition to the 50,000 droplet barcodes, you can resolve all of your data. And in this particular data set, we got 100,000 cells out. So again, if you think about scale, the standard 10x advertised throughput is 5,000 to 10,000 cells. We can now get 100,000 cells in it out of a single experiment, and you can identify all of the expected populations. So this is now cytometry by sequencing. We actually then used the same 28-plex antibody panel and did a CyTOF in collaboration with Matt Spitzer's lab. And we observed that qualitatively, the CyTOF data and the CytoSeq data are in fact very similar to each other, which gives us confidence that we're capturing surface protein expression that's comparable to cytometry. But what's really nice, though, about sequencing is that it's so flexible and customizable to other types of multimodal profiling as well as perturbation experiments. So let me just give you a quick example. We can engineer this secondary oligo that we hybridize onto the antibody-specific DNA molecule however we want. In fact, we could modify this particular end of that sequence to make it compatible with either a 3' 10x chemistry or a 5' 10x chemistry or the 10x ataxi chemistry to get multimodal data. Something else we can do is we can modify the antibody panel to get it compatible with commercially available antibody panels. And so, for example, we followed up with our initial 28-plex data set to make a 60-plex data set. These are all made internally by hand. But then we also work with BioLegend, which has a commercially available 165-plex antibody panel. And we just engineer the hybridization sequence to be compatible with that and use that to profile cells. In both those experiments, we pushed the throughput even more, getting almost 200,000 cells out of a single run of the 10x instrument. Again, the UMaps look great, but importantly, the data is also very quantitative. So in these two experiments, we use the same 10 donors, and we can actually correlate the estimates of cell composition, which are the panels on top, as well as the cell type specific expression of certain surface markers. So CD19 expression in B cells, CD4 expression in T cells, and CD8 expression in killer T cells. And in both cases, we observed high concordance. So we can capture using CytoSeq inter-individual variability in composition in cell type specific surface protein expression. The other, I mentioned earlier that this approach is very flexible. We can also combine this with RNA profiling by adopting the Sci-Fi protocol that was also published in Nature Methods early in 2021. And here's just a pilot experiment where we can get now simultaneously very high throughput by sequencing 5 cell lines, RNA sequencing data, as well as surface protein data, and it's somewhat congruent with each other. So that's CytoSeq. The second quick vignette is about a method called XYZeq, which is using the same combinatorial indexing idea, but now to use that first round of barcoding to encode some additional information. So what is the additional information we're going to encode? Well, it's going to be spatial information. And I hope that it's not going to take too much convincing that understanding the spatial distribution of cells is really important. And that when you do single cell sequencing, you basically destroy all of the spatial information when you take cells and create single cell suspensions. And so even though we make these beautiful looking UMaps or TSNEs, they actually have no spatial information in them. So our idea is to use combinatorial indexing, but use that first index to encode an additional orthogonal label, here spatial location. So here's how we do it. We make these hexagonal wells that are either 500 micron in space, or we can also go down to 200 microns. On the right, I'm just showing a version of that 500 micron microwell slide. This is about 1/3 of the size of the pathology slide. And then what we're going to do is we're going to take a piece of tissue, slice it really thin, about 30 microns, after it's been cryopreserved and cryostat the tissue. And then we digest that tissue onto our 768 well plate. And each well is going to be labeled with a DNA molecule that also serves as a primer for reverse transcription. Critically, the cells will remain intact. We'll keep the cell membrane intact, but permeabilized, and introduce these spatially labeled RT primers into the cell and performing in situ reverse transcription and cell. Because the cell membranes are intact, we can pipette the cells back up, redistribute them into a second a set of wells, and then add a second barcode. And similar to CytoSeq, that combination of barcodes will give you a unique barcode for each cell. And the output of the system is that you get bona fide single cell sequencing data, but importantly, for each cell, we can now resolve where that cell is within the resolution of these spatial wells, which currently is 500 microns. So does this work? So here is a mouse model that we collaborated with Jeff Bluestone on. It's a liver that we've injected in MC38 tumor. If you take a slice and do standard H&E stain, the liver stains nicely light, and then the tumor stains dark. And here's a slice that we took and digested and performed XYZ on our wells. So you can see these hexagonal wells in the background. If you now analyze the data, we can get real single cell sequencing data here on the right to the top. The two major populations are liver in red and tumor in yellow. And what's really nice now, and this is really the positive control here, is what I hope you can appreciate is that the hepatocytes, liver cells, overlap with the light stain, and then the yellow MC38 tumor overlaps quite well with the dark tumor stain. So, how is this data useful, especially compared to single cell sequencing? So if we start this with this teasing again, there's actually two populations here, the orange population and the light green population, that are phenotypically very closely related. In fact, we had to really tweak the parameters to get them to cluster out separately. But if you look at these populations in space, you can see that they're quite strikingly differentially located. So macrophages, or these yellow population, or this, sorry, light green population, co-localizes with MC38 tumor. And so I mentioned earlier, these are macrophages. These are in fact tumor-infiltrating macrophages. And then the orange population co-localizes with the liver. And if you look at some of these markers, they're liver-resident macrophages-like cells or Cooper cells. So that makes a lot of sense. And so this information comes out really easily with the spatial sequencing, but takes quite a bit of tweaking if you look at the single-cell RNA sequencing. The second advantage now compared to other spatial transcriptomic approaches is that the effective resolution of most single spatial transcriptomic approaches is on the order of hundreds of microns because of low capture efficiency. And so you may be able to detect changes in gene expression across space. So for example, here are two different gene modules where the orange module and the green module are both higher expressed in tumor than the liver. But usually that's the end of the analysis, and you need to sort of bring in some orthogonal single-cell RNA sequencing data to figure out or to nominate which cell types might be actually mediating these gene signatures. And with our single-cell sequencing data, we can quickly score these different spatially differentially expressed gene sets and identify, for example, that tumor response module is only expressed by a subset of MC38 tumor cells, and that this immune regulation module is expressed by a subset of the tumor-infiltrating macrophages. And so we think by combining single-cell sequencing data and spatial transcriptomics in a single assay, we now have an unprecedented opportunity to look at co-localizations of cells, but also cell-cell dependencies in transcriptional states. So with that, I'll just summarize some of the stuff that I've showed you guys. I started by talking about this new approach we developed in 2018 called MUC-seq, and show some new results on the application of MUC-seq to interrogate a complex autoimmune disease at the population scale. We think MUC-seq is an unprecedented opportunity to capture variation in composition and cell state. We show that using the approach, we can infer this type 1 interferon response in monocytes and correlate that with naive CD4 decrease in CD4 T cells. We also showed that there's a population of CD8 positive, granzyme H positive T cells that are clonally expanded and transcriptionally heterogeneous. And that if you incorporate in or integrate in genetic information, now you can do unbiased cell type specific cis-UQTL mapping, which we think are much more powerful in annotating disease-associated variants. And then I'm very excited about CytoSeq and XYZ. So CytoSeq allows us to use droplet-based sequencing to now profile surface proteins for hundreds of thousands of cells. So what are we doing in my lab with that approach is now we can do the genome scale perturb-seq experiments where we knock out or overexpress every single gene in the human genome and observe the consequences of those genetic perturbations on surface expression of immune cells, initially starting with T cells. With XYZ, we can now get spatially resolved single-cell sequencing data by keeping single cells intact. We realize a couple of different advantages. So it gives you, I think, more informative data. But going forward, we can also get multimodality, simultaneously get immune repertoire sequencing, as well as CRISPR genome editing as barcodes from the single-cell sequencing. So now imagine experiments where we can take edited T cells, put it into a mouse, and then sequence the tissue with XYZ to see where edited T cells are actually going. And so we're super excited about what we can do with this, and happy to now answer questions about any of these technologies. And I'll just leave my acknowledgement slide up and quick pitch that we are hiring postdocs.
Thanks, Jimmie, for the beautiful talk and sharing with us this exciting and really, really impressive work. So now we open the floor to questions. Okay, I see a question in the chat by Lev. Okay, so shall I just read? I'll just read these out to you. In the SLE story, it looks like the percent myeloid cells was consistently lower in your sequencing data compared to the EHR data, while the percent of lymphocytes were quite similar. Any thoughts?
Yeah, so we have a few thoughts on that. Like one of them is we don't know if like, we might like certain myeloid cells may not have made it through cryopreservation for whatever reason. And the other thing is that like the way that we group myeloid cells, we play with this quite a bit. Like, are the plasmacytoid dendritic cells we're seeing actually myeloid cells? Is that what's been captured when you do a CBC? So there's some, I think we're not exactly sure what we're getting out of the CBC actually. And so, it'd be interesting to, for example, like look at site-seq data where you actually have surface protein expression to potentially do a better job of compositional estimates. But important for all the stuff we do downstream, I think you're right that it looks like it's consistent a little bit lower, but the correlation is still quite good. So we think we're capturing inter-individual variation still in like the same direction that where the real effect is.
Okay, the next question is from Carl Keat. Do you think that single cell technologies will be broadly useful for identifying determinants of drug response or non-response in autoimmune diseases?
No, I think it's possible, except to say, like, I think so much of what's happening in autoimmune disease is happening in tissue. And I'm not convinced that every cell in tissue will eventually make its way to circulation. So one is, we need higher throughput, especially for some of the cells that may actually exit tissue, they could be quite rare to observe in blood. So I think like that's probably the biggest caveat, is that whatever we see in blood will be a proxy of what's happening in diseased tissue. But, I think there is quite a bit we could see systemically in terms of, like maybe some side effects of drugs. And so I think that is, I'm pretty convinced that you should be able to see that in circulation. And whether or not that's going to be useful for understanding side effects, I don't know. But yeah, it's exciting to think about.
Maybe I have a more general question. So in the first part of your talk, especially, you developed this framework and collected this data where you have immune cells, single cells across a very large cohort. And the goal is to understand genetic effects on cell type level expression, and also, the effects of the cell type expression in lupus. So I see that your analysis so far focus on separating out the different cell types and then looking at for each, cell type specific effects versus general effects. And you have that equation where you tease apart the effects into the general term and then the cell type specific term. My question is, since you have single cell data and with your data for each individual, you actually can see the distribution of gene expression across cells within a given cell type. And much more richer than that, you can look at the joint distribution across cells, this variance. Have you guys looked at the changes in distribution that goes beyond the mean? And how important are these changes in distribution that may not be captured by mean? How important is that towards the phenotype you're studying?
Yeah, that's a great question. So I don't know how important they are. We are thinking about ways to even sort of like measure this. I think the first thing we've been thinking about is just like looking at the expression of, this is still at a pseudo bulk level, right? Like whether or not there is, I think what you're asking is sort of like looking at the actual covariance structure, right? We haven't looked at that at all. And I have to think about exactly how one would do that. But we have, for example, thought about like, could you look at mean expression in a particular cell type across individuals and compare that to the expression of a different cell type? Is there, like people have used this, for example, for receptor ligand identification. And that I think is potentially more useful than the current ways of doing receptor ligand analysis from single cell data, which is just looking at overexpression of candidate receptor ligands. But I think thinking about dependencies between cell types, whether or not at the mean level or variance and covariance, is of a lot of interest. And the latter, these sort of like second moments, we need to probably think a little bit more about.
I see another question in the chat from Babak. Babak asked, if I remember correctly, the spatial resolution of XYZ-seq was 500 micrometers. Would you comment on how would you resolve cells within each spatial unit?
Yeah, that's a great question. So, and the answer is going to be a little bit nuanced. I'm happy to follow up with this. So I think 500 is a little low, and we're working on ways to get higher. But I don't think it's critical to resolve the spatial location of every single cell. What we would like to do is have an assay, and this is by design, right? So 500 microns is roughly, we sequence anywhere from sort of like 100 to a couple hundred cells in a 500 micron well. But we just want to be able to identify neighborhoods of cells, right? So we do have single cell transcriptome, but we don't have for each cell where it's localized within that 500 micron space. But we have many 500 micron wells. So we can start asking, what is the dependency between the transcripts of cells that co-localize together in one 500 micron well compared to the transcriptome of two cells that don't co-localize together in a different set of 500 micron wells. And I think like that information is potentially more useful in understanding cell-cell interactions and cell-cell non-sort of, just like non-determined, like basically dependencies across cells that may not even be direct cell-cell interactions than actually having physical location of each cell or even subcellular resolutions. I think like for cell-cell interactions, having neighborhoods of cells is pretty useful. And again, like 500 microns might be a little bit on the low side, and we are trying to increase that to about 200 microns to 100 microns.
Any other questions? All right, if there's no other questions, let's thank our speaker again. And Jimmie, hopefully next time you give a talk, we can invite you over in person. Thank you.
Thanks again for the invitation. Yeah, and yeah, would love to come in person next time.
All right. Okay. Thanks, everyone.
Bye.
Bye.